Good morning to all of you. I hope I would.
Yes.
Yes. Great. So let me start the session because Dr. SNEHASIS has already joined the meeting. Few candidates are still left to join, but we will start. So I welcome Dr. SNEHASIS Daschakraborty for giving us an invite lecture over in the session. Welcome to the first international conference or workshop on computation chemistry using the Gaussian software. Dr. SNEHASIS Daschakraborty, can you please hear me? Am I audible to you?
Yes, I can hear you, Dr. Aggarwal. I can hear you. Thank you.
Let me do a brief introduction about Dr. SNEHASIS Daschakraborty. Sir has completed from SN Bose Center for National Center for Basic Sciences, Kolkata. Followed by he has joined Department of Chemistry, IIT Patna as an assistant professor in 2016. His major research interest is to investigate the structure and the dynamics of various types of liquid involving supercooled water, ionic liquid, and many more types, and the biomolecules as well. In his research group, they go for the computer simulation as a prime tool for the investigation. Their primary objective is to build a connection between various experimentally observed properties of various complex liquid and biomolecules with structure and the dynamics. To his credit, there are more than 30 publications of international repute. Major awards are also in his credit, involving Young Scientist INSA and many more, to just count a few. So with that, now I would like to hand over the session to Dr. To take up the lecture. Sir, over to you.
Okay, so can I share my screen?
Sure, sir.
Yeah. My screen is visible?
Just a second, sir. Yeah. It is visible to, let me confirm from the participant. The slides are visible to all of you. The session now.
Okay.
Can you make it on a presentation mode?
Yeah.
Great.
It is not on a presentation mode. Can you just make it that one? Yes, now it is visible properly.
I have made it full screen. Is it visible to you? This is, I think, in presentation mode. Is it visible?
I think it's fine. We can go ahead with this. It's clearly visible to all of you. Am I correct?
And I made it full screen also. Okay, if it is not in full screen, I will try it again.
Sir, there will be a way to share your entire screen. There will be a way to share your entire screen.
Yes?
There is a way to share your entire screen. If you click on that upward arrow in the panel, it will share your entire screen. I think it's
inter screen. I'm sharing inter screen only.
So we can start the session now. Yeah, please.
Okay, let me stop again and do the whole screen. Okay, so I think here mostly students, right, Dr. Aggarwal?
Faculties are also there from different institutes.
Okay. Very good. So I think now the full screen is visible, right?
Correct. It's visible to all. I request to all the participants to maximize their screen.
Okay. Good morning, everybody. And I especially thank Dr. Aggarwal for giving me the opportunity in this conference.
Sorry
Okay.
for the disturbance. There was.
Yeah, no problem, no problem. So, good morning, everybody. And I thank Dr. Aggarwal for giving me the opportunity to present our work, the kind of work we are doing at IIT Patna for the last 5.5 years. And although I have seen the theme of this workshop is mostly density functional theorem based, but this is another side of the story for computational chemistry or computational physics, even computational material science also. That is molecular simulations using classical techniques, classical force field. So this is one of the areas we are working at IIT Patna with my research group, and that works actually involves classical molecular dynamic simulation technique. So basically, the title of my talk is Adaptation of Cell Membrane in Extremophilic Organisms. So this problem we have started back 2018. So this is my lab group page. You can see directly going to that. This is my email address. If anybody has any questions about the work we are doing, about papers, you want to access papers, I can directly send also. So molecular simulation lab at IIT Patna, my lab, are basically involved in 4 different areas. One is supercooled water, particularly breakdown of Stokes-Einstein relation, and how Stokes-Einstein relation is a relation which connects the diffusion and viscosity, and how that breaks down at supercooled water. Supercooled water is the water below freezing point, because water freezes at 0 degrees Celsius at atmospheric pressure. So if you reduce the temperature below 0 degree, you will the water will still can be kept as liquid, and that state is called supercooled water. And in that supercooled water, there are tremendous amounts of anomalous behavior are observed experimentally. One such anomalous behavior is Stokes-Einstein breakdown. Stokes-Einstein relation is basically from the name as suggests. It comes from the research of Stokes and Einstein's Brownian motion. When you connect them, diffusion coefficients of molecules in liquid state basically is coupled to the viscosity of the medium. If viscosity is higher, viscosity, the flow, the fluidity, right? So if viscosity increases, then diffusion of the molecule decreases. So that actually that is given by the Stokes-Einstein relation. But sometimes, particularly at supercooled conditions, this Stokes-Einstein relation breaks down, and we have given a new theory of it, new approach to explain this breakdown of Stokes-Einstein relation by translational germ diffusion approach. Then we, as a new student, joined in 2018. We started working on model cell membrane. We know cell membrane is made of lipid bilayer and proteins. Lipid membranes we are mainly focused on. We are focusing in extremophilic organisms. Extremophilic organisms, I will go into this in details. This is the topic of of today's talk. Then nanoparticle cell membrane interactions also we are working on, where different nanoparticles like carbon nanotube, graphene sheets, carbon dot, etc., and also added both urea and TMAO at different concentrations, particularly 10 weight percentage. And we have seen how this urea or TMAO, how they actually affect or influence their the phase transition of the lipid membrane because of this low hydration conditions. So what we are seeing here, these are the two properties we have also shown previously, area per lipid and thickness. These are the packing density parameter of the cell membrane. You can see how as the hydration level decreased from 35 to 4, you can see this is where there is no, it's a blank system where no urea or TMAO was added. You see, it's a sharp decrease of the area per lipid and sharp increase of the thickness. And the increase or decrease such that it almost reaches the gel phase. Okay. But then, as we added a TMAO, a trimethylamine oxide, we can see the increase, decrease is extremely sharp. Means TMAO is basically what it is doing, it is accelerating the phase transition. It is inducing the phase transition. So it is bad actually for this dehydration-induced situation. But what is the role of urea? This is blue color lines you can see, that is for the urea. As urea is present, you can see that reduction of the area per lipid or the increase of the area per lipid is significantly decreased. So almost intact, you can say the change is almost not less than 0.58 nanometer square, or thickness not more than 3.7 or 3.8, which indicates that this phase transition was completely stopped by the presence of urea, and that urea is present in the cells of the desert microorganisms. So we can see different other parameter also, the similar behavior, that as urea is present, it actually does not allow it to have phase transition from fluid to gel phase. So now in the second problem, we have, and I should definitely say that in this problem, how this TMAO does it, because, urea does it, because urea has a strong hydrogen bond site, okay? And urea directly interacts with these lipid molecules, and thereby it acts as water, okay? Although water is not present, but looks like water, same role of water is done by urea. But TMAO, completely opposite. TMAO is actually goes out of the membrane and it draws some more water from the cell membrane. So further dehydration process occurs. Okay. So some deep osmolytes, TMAO is called trimethylamine oxides is actually called deep osmolytes. Urea is also called deep osmolytes. What is the role of these deep osmolytes at high pressure induced phase transition? You know, this as I was saying, this is a nature paper, Nature Ecology and Evolution. The morphology of genome and snail fish from the Mariana Trench provides insights into deep sea adaptation. In this Mariana Trench, which is approximately 6,000 meters, okay, from the sea level, lots of snail fishes are present. Pressure is extremely high. And while it was dissecting this snail fish, it was present, TMAO was present, trimethylamine oxides are present in an excess amount. Question is, what is the role? Is there any role for these osmolytes for the cell membranes? For protein, it was clearly visible that this TMAO has a significant stabilizing effect on protein. But what about cell membrane? It was not studied. So we have another paper which also shows this TMAO deep sea osmolytes on the temperature and pressure dependent structures and phase behavior of lipid membrane by Winter's group. So recently in 2019 it was published. So by these works we motivated to work on this problem. So we have artificially made high pressure regions. We change the pressure from, you can see, from 0 bar or 1 bar to 2000 bar. 2000 bar is extremely high. Almost a similar pressure at the Mariana Trench. And we can see there is a phase transition of the cell membrane as the pressure increased. You can see there is a sharp change from here to here as the pressure increased from 400 to 800 bar. We can say 800 bar is the roughly the phase transition pressure. So this urea in TMAO does anything on this? Unfortunately, we do not see any role of TMAO in urea on this pressure-induced phase transitions. At a smaller pressure, I can see some change. This blue color is for the urea phase. So urea somehow stabilize the membrane at low pressure, but at high pressure, this phase transition cannot be stopped by urea or TMAO. So that paper also published in JPCB. Conclusion is osmolytes have large impact on fluid phase transition, particularly for the dehydration-induced phase transition, not for the pressure-induced phase transitions. And the main important cause for this stabilization is the strong hydrogen bond interactions, okay, which urea has, but TMAO cannot interact with lipid molecules strongly, that we have seen from our simulation studies. And TMAO also gets excluded from the membrane and dehydrates further, which actually accelerates or induces the fluid to gel phase transitions. Okay, so I acknowledge different funding agencies, ACRB Early Career Research Awards, CSR, DST Indo-Korea Joint Program, DST Inspire Fellowships, and also my student Sakira and Urchita, who have done this lipid membrane work. Because Shivam has done this, and Ghulam also is working on the supercooled water problems, and Tanmoy is on the amphiphilic, the absorption of amphiphils on water surface. And thank you all for your time.
Thank you, sir. It was a very splendid lecture, I would say. Rather, in this session, I correlate. Am I audible to all?
Yes, I can hear you.
So during the session, I could rather correlate what we studied in our class 11th, 12th, then moving on to the basic standards, moving on to the research field. I can correlate everything. So it started from what are the extremophiles, the role of these extremophiles, how they survive in different acidic, basic, high temperature, low temperature, pressure, extreme condition. But it never came to our, or rather my brain, like how they changes their lipid bilayer structure for their survival. Brilliant studies, computationally mimicking them, another branch of a science. The part which I like the most, it was the addition of so many functionalities like a urea or side moieties, which significantly impart their phase transition or their changes in the structure. Brilliant study, very much impressive. So I would like to say thanks to Dr. SNEHASIS for giving us a plenty of our time and a very valuable information to all of us. I would like to have my candidate or my participant, if they have questions to SNEHASIS.
Yeah, okay. Thank you, Dr. Aggarwal. So I can see, sir, I would like to know which computational method. Okay, so this is done using molecular dynamic simulations. Molecular dynamic simulation is another part of these computational techniques. Like in DFT, you have used, I think in this workshop also, you are learning how this computational, like these ab initio calculations, like quantum chemistry is helping to understand their molecular structures, optimization, geometry, etc. So using that geometry, you can see, you can tell that molecular dynamic simulation starts from the ab initio charge of different atoms. So using the charges of the atoms, different bond lengths, dihedral angles, you can basically model a molecule. Once you model a molecule, now you can take many molecules, because now it is no longer a computational chemistry calculations. So it is computationally less expensive. So now you can take many molecules together and you can use Newton equations, firstly calculate the force, and then using the force, you can put it in the Newton equations to solve the molecular trajectories. Okay, so how the molecules are moving inside a box, you can actually see it. And using the molecular trajectory, or you can say phase space trajectory, you can solve any parameter, and you can calculate any quantities you want using the statistical mechanical approach. Okay. So basically, it is molecular dynamic simulation in one sentence. Computer simulations. Different packages are also available. Like in DFT calculation, you have Gaussian. So similarly, in molecular dynamics, you have GROMACS, you have LAMMPS, etc. Hope I answered this.
we essentially go with the GROMACS for all these kind of molecular dynamics studies, different methods, and then we get out our next question, please, from the participant. We can have a 2 quick question because sir has to leave for the class. Why you choose DFT studies, not HF, MP2, and PCSD? Ashok, please think of the kind of a system sir is already working on. They are the giant macroscopic molecules. And if you recall, yesterday we had a discussion for these giant macroscopic molecule, DFT will not be able to yield you the result unless or until you have a very big supercomputer or not less than a supercomputer. And even then it will fail. So molecular dynamics, which is a little less relaxed criteria, we have learned in the yesterday session, it relies on the Newton mechanics. So little relaxed criteria, you can get the result easier as compared to the DFT study. I hope, Ashok, this would have answered your question. Next question, please. We can have some 1 or 2 quick questions because sir has to leave for the class. Great. So with that, I again thank Dr. SNEHASIS for joining us and delivering us a very wonderful lecture. I thoroughly enjoyed it from the very starting towards the end. DFT is for 1,000 atoms, but dear, even if let us say it is applicable to, let us say, 1,000 atoms, 5 layers, molecular layers, or a kind of a lipid, sir is already working on more than 10,000.
Almost like 70,000 atoms, you can say.
Correct.
In the cell membrane, you will have 70,000 atoms, which is almost impossible. Yes.
Impossible for the DFT study. Correct.
Yes.
True. Your PPT, fine. So, I mean, it can be extended to the molecular dynamic studies only. Molecular quantum mechanical studies, it will dramatically fail. Great. So with that, I'll thank Dr. SNEHASIS for delivering us a very wonderful lecture and giving us a valuable time. From the audience and the participant side, I'll thank you for joining us.
Thank you.
With that,
thank you, Dr. Aggarwal. Thank you. Namaste. Thank you. Bye. I can log out. Huh?
You can. My participant would like to also thank you.
Thank you very much. I hope you are thoroughly enjoying the workshop.
From the very beginning, I enjoy it like anything because generally these kind of webinars, people get bored. But in your session, enjoyable and informative as well. Very informative. Great.
Thank you.
Thank you for giving us the time. So, sir, now you can take up the class. I know you have a very busy schedule. Thanks for giving us an extra time.
Thank you very much. I actually cannot log out. Can you please expel me? Actually, I cannot log out.
I cannot expel you out at all. I'll rather, it's removed. Fine. Just a second. Remove. Remove. Yeah. So candidates, participants, I hope all of you would have enjoyed the session. See, basically what sir has discussed, I'll take just 5 to 10 minutes, then we'll start with the training part of today. So what basically sir was discussing in the session, he is working on different types of lipid bilayers. Why he's working on lipid bilayers? Because as he said, there are different types of extremophiles, like we have the methanogens, acidophile, basidophiles, and then we have so many kind of bacteria and viruses which live in various acidic condition, basic condition, high pressure, low temperature. So how do they basically survive? They survive by changing their membrane structure. So basically his research focus is to study how the lipid bilayer structure or membrane structure, they changes with the application of extreme condition. Very good branch of our science, mainly focusing towards the application part. I would rather say that all of you go through his homepage, web page, and just after your training get over, see, I can see where are the scopes after the training you can apply to him. So I can see that his research interest is to investigate at the molecular dynamic level, but his research focus is not towards the study of the interaction between the two molecules which are aligning at a certain specific angle. Give us a study. Like as an example, he emphasized when you increase the temperature, at that particular increasing temperature, you need a very strong bilayer. So if you need a very strong bilayer, may I ask one question? If you need a very strong bilayer, in that case, the interaction between the molecules which are present in the layer, they should be high or they should be low. If you need a very rigid bilayer, Answer in the chat box. If we need a very strong bilayer, in that case, we need a strong interaction between the molecules or weak interaction between the molecules. Answer it quickly, maybe vocally or in the chat box. Imagine, exactly, we need a strong interaction. And see, what we all know, there are 4 or 5 types of, see, I'll take up like a BSc student. I assume that I am a BSc student of the lecture right now. So now assume that you have 4 or 5 types of major attraction. Ionic interaction, covalent interaction, dipole-dipole interaction, hydrogen bonding. As far as I know, the hydrogen bonding will be the predominant when you will take up the bilayer structure. So now, if let us say you will align the 2 molecules in a certain fashion so that it will maximize intermolecular hydrogen bonding, the layer will be dramatically stabilized. So these kind of studies can be very easily investigated using your DFT model. A plus point, certainly he will be looking for some person who can carry out the DFT studies, as he has already mentioned in the 3rd article. So I emphasize that all of you learn the DFT studies, and as soon as your training is over, specifically the BSc and the MSc students, write to him that you have now learned the DFT studies into the Gaussian and would like to collab or would like to join his research group. He will definitely ask what kind of a proposal you have it with you. So you can mention that you would like to study the interaction between the 2 molecules, adjacent molecules in a layer. You would like to tune the structure of the molecule so that the interaction between the 2 adjacent molecules will increase and dramatically increase. So these kind of proposals can be submitted. I hope it would be clear a little bit more now. I don't want the session to be boring, but I hope it would be clear to all of you now what Sir was trying to emphasize. Clear to all of you? So with that, I'll thank all of you. Now I'll give a time of some 9 minutes. It's 11:51. Fine. So it's still 11:51. I'll give a time of some 5 to 6 minutes. Please join back using the same meeting link at 12 o'clock. We'll start with the Gaussian training. Okay. Or you can stay over here, but I need to stop the recording. Thanks to all of you. And see, hydrogen bonding helps in the soluble the compound in a water. If lipid will show, then lipid, and I lost the. Yeah, the question was, the question was from the Dr. Ashok, hydrogen bonding helps in the soluble the binding compounds in the water. If lipid will show, then lipid can soluble. So therefore, see, that is the task of a research. You have to tune the molecular structure in such a way that it will maximize the hydrogen bonding, but it will not get solubilized. So maybe you will have to put some functionalities over there which will be hydrophobic in nature, more hydrophobic in nature. And also you need to put up a substituent in such a way that the specific angle between the 2 molecules should be controlled. So this is the area of our research that you will learn it more when you will be into this particular field. Great. So take up a time of what is the logic. I could not understand that as lipid is hydrophobic. Just a second. I lost the question. What is the logic that I could not understand that as lipid is hydrophobic, sir? See, lipid is basically hydrophobic in nature. As sir has also emphasized, lipids are basically hydrophobic in nature. So if they are hydrophobic in nature, that means they would like to stay away from the water molecule. Lipid will show hydrogen bonding. See, we don't want the lipid to show the hydrogen bonding with a water molecule. We want the lipid to show the hydrogen bonding between the 2 adjacent molecules. Now, how it can be achieved? See, imagine you have, I'll take up a simple example. If let us say, I'll take an example of paranitroaniline, a very simple molecule. Tell me, what sort of a intermolecular, what sort of a bonding will be predominant? Is it going to be intermolecular hydrogen bonding or intramolecular hydrogen bonding? If I have a paranitroaniline, a very simple case. Imagine you have a nitro group on
Inter.
Exactly. It will be
Inter.
Similarly, now instead of, let us say, I'll take orthonitrophenol instead of a paranitrophenol. If I'll take a orthonitrophenol, the tuning will be like intra. Ashok, you said incorrectly first. If you have a para, it will be intermolecular. Yeah. So where I was, so for the, if let us say you would like to increase the intramolecular hydrogen bonding, you need ortho substituent. In this particular case, if you want inter, you need a para. So like that, you have to tune your among themselves. and that is a very frontier area of a research. If you will look at the literature, you will find enormous number of molecules wherein people look at how to modify the chemical substituent to get the desirable, controlled, tuned lipid structure. Very frontier research area. Great. I hope, Ashok, this would have answered your question. Maybe to some limit, if not completely. Well, lipid show hydrogen bonding, sir. See, lipid, it's not just like a 2-atom or 4-atom or 6-atom molecule. You need to put up a certain functionalities over there, maybe some 2 or 3 functional groups, which will initiate the intermolecular hydrogen bonding. The entire molecule may not show the intermolecular hydrogen bonding, but at least some part of that molecule will show the intermolecular hydrogen bonding. I just gave a very one type of an interaction. Maybe you can think of the pi-pi interaction. Long chain of fatty acid will not be able to. Maybe you have the carboxylic acid group. So carboxylic acid group, maybe if you will align the molecule in such a way that there will be an intermolecular hydrogen bonding, it can be tuned up. So there are various ways which you can think of. Some chemical modifications can be done so that at high temperature or low temperature, the lipid structure will undergo some kind of a chemical reaction, an equilibrium kind of a reaction, so that at a low temperature, this equilibrium will be more towards the reactant or product side, and that structure will be more stabilized. So many possibilities can be think of. I would say it's a frontier area of a research. Think about it and submit a proposal to him as early as possible, maybe for the collaboration or maybe for your BSc and MSc project, because he's looking for BSc and MSc project students, PhD positions and postdoc. He joined Patna in 2016. So looking to expand his research group like anyway. And tomorrow we are having a session by Professor T.P. Radhakrishnan. I would say he's one of the marvelous person in terms of nanoparticle chemistry.